So this is chapter 20, the heart. This is our second chapter in the cardiovascular system. And we'll start with a brief introduction. So the heart is actually two pumps that are working side by side, and it's important to realize that blood does not flow directly from the left side of the heart to the right side, nor does it go from the right side to the left side. So they are two completely separate standalone pumps. The left side is a pump that pumps blood out to the systemic circuit. The systemic circuit is going to be all of the vessels that are outside of the lung. So anything that is non-lung would be part of the systemic circuit. And the right side of the heart pumps blood to the pulmonary circuit, which is just a circuit that includes the lungs. So we are going to go over blood flow through the heart and body in a later section, and you are going to need to know those structures in order, both for lab and lecture. It's very important that when you have made it through A&P2 that you can recite the blood flow through the heart and body. I also want to introduce you to some terminology related to the vessels, and then we're going to look at vessels in detail in chapter 21. So arteries are vessels that take blood away from the heart. So you can remember that they both start with the letter A, so arteries go away from the heart. Capillaries are where exchange takes place between the blood and the cells of your tissues. And then veins are bringing blood back to the heart. So arteries are going away from the heart, veins are coming back to the heart. Also, when we look at these various images in the textbook, and then when you look at models in the lab, you should also note that the colors have a meaning. When you see red, this indicates a blood vessel that is carrying oxygenated blood, meaning that the hemoglobin is saturated with oxygen. And where you see blue or purple, this represents deoxygenated blood. And as we're going to see later, there still is some oxygen in the hemoglobin, but a lot of it is also gone as well. So the cells of the body have taken that oxygen away from the hemoglobin and are using it in their mitochondria to make ATP. So we call that deoxygenated blood. Now, you may have heard that there's a general rule that arteries are red and veins are blue, but that shouldn't be your defining characteristic of them. So yes, while most arteries are going to carry oxygenated blood, there are exceptions. And while most veins are going to carry deoxygenated blood, there are exceptions. So it's better to learn that arteries are carrying blood away from the heart, veins are carrying blood back to the heart. Red means oxygenated blood, and blue or purple means deoxygenated blood. And if you remember those things, then you'll always get it right. So the heart is a tough little organ. It actually beats about 100,000 times every day and pumps about 8,000 liters of blood per day. And I like your textbook gave you a little analogy. It said, imagine using your turkey baster to try to fill up a gallon jug full of water, and you would have an idea of just how hard the heart works every day to pump all of this fluid. So we're going to start with the location of the heart and a discussion on the pericardial membranes. So the heart sits in the thoracic cavity. You learned in A&P1 that the thoracic cavity sits above the diaphragm. Below the diaphragm was the abdominopelvic cavity. And within the thoracic cavity, the heart sits directly posterior to the sternum. So in this cut version here, you can see the sternum at the very front, and then the heart is going to sit right behind it. Also note that the esophagus runs right behind the heart, which is why when you have acid reflux and other problems where the stomach acid come up into the esophagus, we call it heartburn, because it can actually feel like the pain is in your heart, but it's really sitting right behind your heart. The heart is located in the anterior portion of the mediastinum. So we introduced the mediastinum to you in A&P1 as well. So we talked about how the thoracic cavity had the pericardial cavity, the two pleural cavities, and then it had this region called the mediastinum where you had the big blood vessels, the esophagus, and the trachea all run through this area. The heart also sits a little left to the midline. So if you look at a position of the heart in relation to the rib cage, you can see that it is slightly off-centered, so it is slightly to the left side. And just like we saw last semester with the sacrum, the base of an organ doesn't always mean the bottom. The base is always the widest part of an organ, and so the widest part of the heart is at the top where the large vessels come out of the heart. So the base is actually on the top of the heart, and then the apex is the pointed part at the bottom, and the apex of the heart is going to sit even farther away from the midline, as you can see from this image at the bottom. So also as a review from last semester and chapter 1, when we introduced you to these body cavities and the membranes that line them, the pericardial cavity is one of those body's major cavities, and it's found within the thoracic cavity. And there are two membranes in each of these cavities, but we're going to focus on the pericardial cavity. So the membrane called the visceral pericardium is the membrane that is going to line the surface of the heart itself. So it is actually on the surface of the heart. The parietal pericardium is going to be the membrane that lines the outside of the cavity. And then there is this space between the two membranes. It is this space that is actually called the pericardial cavity, and this space is filled with fluid. And both of these membranes are called serous membranes, which was a type of membrane we introduced you to in chapter 4. So this space is full of fluid, and it's called pericardial fluid. So pericardial fluid is the fluid inside the pericardial cavity, and the function of this fluid is to reduce friction as the heart beats. So when the heart beats, these two membranes are going to rub up against each other, and that friction is reduced by the fluid. And again, as we mentioned last semester, you can kind of picture this as if you had a hand that you stuck into a balloon. And so the air would represent the space within the cavity where you would find that fluid. The part that's touching your fist would be the visceral layer, and the part that's on the outside here would be the parietal layer of the pericardium. So that should be review for you from A&P 1. So to go into a little bit more detail with the pericardial cavity, the visceral pericardium, which again is the membrane that is attached to the heart itself, can also be called the epicardium. The parietal pericardium, which is what is lining the cavity, and you can see in this close-up view here, just like all serous membranes, it has a thin layer of mesothelium, and then underneath that is areolar tissue. But in the case of the parietal pericardium, there is also another layer of dense collagen fibers that form a thicker layer called the fibrous pericardium that is then going to surround this entire pericardial sac. Pericarditis is infection and inflammation of the pericardial membranes. This can result in an increased fluid production of the pericardial fluid. And cardiac tamponade is the condition when the movements of the heart is restricted due to excess fluid in the pericardial cavity. So the fluid really has nowhere to go, so if it builds up in this cavity, it puts pressure backwards on the heart and reduces the space for the heart to move. So this can become rapidly life-threatening. Just FYI, going back to the idea of root words helping you remember things, tampon is a root word that means a plug. So this is actually the fluid is building up and plugging the heart and preventing it from moving. And this can be caused by either pericarditis or bleeding from a wound, so like a stabbing or other type of wound that might cause bleeding within the pericardial cavity. In this first section, where we're going to cover actual heart anatomy, we're going to look at the outer superficial structures and the composition of the heart wall. So we'll start with this picture showing the anterior surface of the heart. So each side of the heart has two chambers, one atrium and one ventricle. And as I mentioned before, each atrium and ventricle work together as a single pump. And just to remind you, the left side is going to pump blood to the systemic circuit, and the right side is going to pump blood to the pulmonary circuit. So when you look at the anterior view of the heart, we can see the right atrium, we can see the right ventricle, and then we can see a little bit of the left ventricle and a little bit of the left atrium. Now when you see these terms on these images that say like auricle of left atrium, the auricle is an expandable extension of an atrium that can be seen externally. And when you do your dissection in lab, you'll see that these kind of look like little flaps. So some other structures to know. The anterior interventricular sulcus is found right here. There's a lot of fat and blood vessels in the sulcus, but it's this groove that runs between the ventricles. So it acts as the boundary between the ventricles on the anterior side. So it's right here in between the right ventricle and the left ventricle. And then there are a lot of vessels that you're going to become intimately familiar with. So to introduce you to these, there is the aorta. The aorta carries oxygenated blood from the left ventricle to the systemic circuit. The aorta has several main branches that we're going to talk about, but not until we get to chapter 21. And we're also going to talk about this ligamentum arteriosum, but that's when we get to the fetal heart that we'll discuss that. The pulmonary trunk and the pulmonary arteries. So in this image, the pulmonary trunk is right here. And then shortly after it leaves the heart, it branches into a left and right pulmonary artery. Only the left pulmonary artery is visible right here. These carry deoxygenated blood from the right ventricle to the pulmonary circuit. And then you have the superior and inferior vena cava. And on this particular image, all we can see right now is the superior vena cava over here. And this is going to carry deoxygenated blood from the systemic circuit back into the right atrium. So now we'll take a look at the posterior side of the heart. So back here, we're going to find a posterior interventricular sulcus, which again is going to be the boundary between the left and right ventricle, but now we're on the posterior side. Also notice when you're looking at the posterior view, the left side is going to match your left side and the right side is going to match your right side. But when you are looking at an anterior view, the left and right are going to be reversed. So notice the right is on your left and the left is on your right. We can also see the coronary sulcus, which again is going to have fat and blood vessels within it, but this is the boundary between the atria at the top and the ventricles at the bottom. You can find the coronary sinus in this area, and we're going to talk about that later when we talk about actual circulation to the heart muscle. So again, major vessels to know. You can see the aorta peeking over the top here. So we already mentioned that it carries oxygenated blood from the left ventricle to the systemic circuit. We can't see the pulmonary trunk, but now you can see the left and right pulmonary arteries that are branching apart right here underneath the aorta. They carry deoxygenated blood from the right ventricle to the pulmonary circuit. Now something that we couldn't see from the front are the left and right pulmonary veins right here on either side of the left atrium. They carry oxygenated blood from the pulmonary circuit back to the left atrium. And then we have the superior and inferior vena cava. Before we could only see the superior one, now we can also see the inferior one. And they carry deoxygenated blood from the systemic circuit back to the right atrium. So now let's take a look at the cross section of the heart wall. So the heart wall is made up of 3 layers. The outermost layer is called the epicardium. And this is the same thing as that visceral pericardium we were talking about. So that's shown right here. It would be this layer right here where it says mesothelium and areolar tissue. This cross section also shows you the cavity where the pericardial fluid will be, and it also shows you that parietal pericardium, which would be lining the outside of the cavity. But because we're just looking at the heart wall, I am only including the visceral pericardium in this list, and it's also known as the epicardium. The middle Oh, and this is a type of serous membrane that we covered back in chapter 4 when we talked about serous membranes, cutaneous membranes, mucous membranes, and synovial membranes. So the middle and fattest layer of the heart wall is called the myocardium. This is where you're actually going to have that cardiac muscle tissue, which again we introduced you to back in chapter 4 when we did tissues, and we're going to go into more detail on the function of the cardiac cells in this chapter as well. And then the innermost layer that would be facing inward to where the chambers of the heart are is called the endocardium. So the endocardium is going to be a simple squamous epithelium called endothelium, and then it's going to have a little bit of areolar tissue underneath that. So this endothelium that lines the inside of the heart is continuous with the endothelium that lines blood vessels, and we started talking about endothelial cells in chapter 19, so you should already be somewhat familiar with that term. If we take the endothelium together with the areolar tissue, that is what makes up the endocardium. So those are the 3 layers of the heart wall. So before we look at the internal anatomy of the heart, let's stop for a minute and talk about the valves and valve problems. So the heart has 2 types of valves, and it has 2 of each type, so it has 4 valves in total. So remember, 4 chambers and 4 valves. So the atrioventricular valves, also called the AV valves, are folds of fibrous tissues between an atrium and a ventricle. And the flaps of the valves, which are also called cusps, are attached to papillary muscles by connective tissue fibers called chordae tendineae. So the papillary muscle functions to maintain tension on those AV valves by pulling on those chordae tendineae, and it prevents them from swinging back into the atria, which is what we would call a prolapse of a valve. So the chordae tendineae are those connective tissue strands that connect the valve flaps to the papillary muscle, and when you do your dissection, you'll see they actually do look like little strings, and this is where the term heartstrings comes from, as in you're tugging on my heartstrings. So the 2 atrioventricular valves are the tricuspid valve on the right side. It's called tricuspid because it has 3 of these flaps, which are also known as cusps. And then you have the bicuspid valve on the left side. And again, cusp is one of those little flaps, and this one only has 2, so it is a bicuspid. This one is also known as the mitral valve, and that is because the name mitral comes from the Latin word mitre, which is a bishop's hat. So a bishop's hat has like these 2 little flaps on the top, and that's actually kind of what the bicuspid valve looks like. So the other type of valve is a semilunar valve. These have 3 half-moon cusps of connective tissue that sit between a ventricle and one of the great vessels. These valves do not have chordae tendineae because they are not exposed to the same pressure as the AV valves. So you have 2 of these. The pulmonary semilunar valve is going to be on the right side at the base of the pulmonary trunk, and the aortic semilunar valve is going to be on the left side at the base of the aorta. So here's a picture from the textbook. So just quickly, I'll point out these 2 types of valves to you. So we're just looking at the left side in this image. So you can see between the left atrium and the left ventricle, we have an example of an atrioventricular or AV valve. And in this particular example, it is the mitral or bicuspid valve. There's its 2 flaps, 1, 2. There are the little strings or the chordae tendineae that are extending from the bottom of the flaps down to where they adhere to papillary muscle. And then up here at the base of the aorta is where we would have the aortic semilunar valve, which are these 3 little half-moon flaps that close up here, and there are no chordae tendineae. But what I want to draw your attention to is on the left side. This is showing the valves from the top view so that you can see both of the AV valves and both of the semilunar valves at one time. And notice that when the heart goes through the cardiac cycle, which we're going to cover in a later section, both AV valves are open or shut at the same time. So if the tricuspid is open, the bicuspid is also going to be open. And conversely, both semilunar valves are open at the same time. So when the aortic valve is open, the pulmonary valve is also open. But also notice that the two sets of valves are never open at the same time. So when the AV valves are open, the semilunar valves are closed. And when the semilunar valves are open, the AV valves are closed. And this will make a little bit more sense to you when we actually get to the blood flow through the heart, and also when we actually look at the cardiac cycle in detail. So bottom line, when the ventricles are relaxed, so both left and right, when the ventricles are relaxed, the AV valves are going to be open and the semilunar valves are going to be closed, and that's shown up here at the top. When the ventricles contract, and again, this will happen on both sides simultaneously, the AV valves will be closed and the semilunar valves will be open. And the heart sounds that you hear, the two audible heart sounds that you can hear very easily, even just by putting your ear to a person's chest, they typically are called lub-dup or lub-dub. And that first sound, the lub, is the sound of the AV valves closing. And that second sound, the dub, is the sound of the semilunar valves closing. So I have a quick little video just to give you an idea of how this works. This video isn't going to last very long. I just want you to see how the valves opening and closing correlate with the sound you hear when your heart beats. Lub-dub, 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 lub-dub. I had my sound on high, so hopefully you were able to hear that. It was still kind of quiet, so I narrated there at the end, but hopefully you get the idea. So the function of the valves is to prevent backflow of blood. And in this image, you can see all four valves. So you have the tricuspid valve on the right side between the atrium and ventricle, the mitral valve or bicuspid on the left side between the atrium and ventricle, the aortic semilunar valve at the base of the aorta, and the pulmonary semilunar valve at the base of the pulmonary trunk. So the AV valves are going to prevent backflow of blood into the atria when the ventricles contract. So the tricuspid valve is preventing backflow from the right ventricle to the right atrium. And the bicuspid or mitral valve is preventing backflow from the left ventricle to the left atrium. The semilunar valves prevent backflow into the ventricles when the ventricles relax. So the pulmonary semilunar valve is preventing backflow from the pulmonary trunk into the right ventricle. And the aortic semilunar valve is preventing backflow from the aorta into the left ventricle. So regurgitation is a term we use to describe when you do have the backflow of blood through a damaged or dysfunctional valve that doesn't close properly. And this can be heard through a stethoscope as a heart murmur. And so here is an example on the right side of where if you have blood that goes back through a valve that should be tightly closed, that is called regurgitation. Prolapse occurs when a valve flops too far backwards past its normal closed position. So if you look on the left side here, you can see the AV valve right here. It's not supposed to go any further back into the atria than this. And on the right side, it shows what happens or what it looks like if you have a valve prolapse. So the valve is extending too far back into the atria, which means that it can't shut properly, which means you end up getting regurgitation. Stenosis is a term we use when a valve becomes rigid or hardened. Usually it's because of atherosclerotic plaque buildup, and it makes it more difficult to open, and it's not as freely movable as it normally should be. The mitral bicuspid valve is the most common valve to have issues, and that's because it's on the left side of the heart with the highest pressures, because the left side of the heart is having to pump out to the entire systemic circuit. And valvular heart disease, or VHD, is when the valve function deteriorates to the point that the heart cannot maintain adequate blood flow, and then this has the potential to lead to heart failure, cardiac arrest, or even death. So in this section, we'll look at the second part of heart anatomy, where we'll talk about the structures on the inside and differences between the right and left sides of the heart. So now let's look at the inner structures using this image from your textbook. So structures you should know are the four chambers. So we have the left atrium, the right atrium, the left ventricle, and the right ventricle. Again, remember that when you're looking at an anterior view of the heart, so when you're looking at it head on, the left and right are reversed to your left and right. You should also know the four valves. So over here we have the bicuspid or mitral valve with the two cusp or flaps. Over here we have the tricuspid valve. Right here at the base of the pulmonary trunk we have the pulmonary semilunar valve, and the aortic valve is not really that visible. It's at the base of the aorta, and just a little bit of it is peeking out right there. So those are the four valves, and then with the atrioventricular valves, you've also got the chordae tendineae, which are the little strings coming down, and they're attaching to these muscles called papillary muscles. You should also know the great vessels, which we covered in the earlier section. So we have the aorta. So you first have the ascending aorta, which goes into the aortic arch, which then turns into the descending aorta. But we're going to cover the different parts of the aorta more in chapter 21, as well as the branches, the initial branches from the aorta. We also have talked about the pulmonary trunk, which is right here. The pulmonary trunk divides into a left and right pulmonary artery, and then the closer you get to the lungs, the left and right pulmonary arteries branch into two left and two right pulmonary arteries. Behind here you can see a bit of the left pulmonary veins going into the left atrium. The right pulmonary veins are hidden in this view. We can also see the superior vena cava over here and the inferior vena cava down here. You should also know the interatrial septum is the separation between the two atria, so between the right and the left atrium. Remember that in an adult heart, blood never flows from the left side to the right side. It does in a fetal heart, which we'll talk about later, but in an adult heart, there is no mixing between the left and the right sides. The interventricular septum is going to be the separation down here between the left and right ventricles. The moderator band is shown right here, and this is only found in the right ventricle, and it's a muscular ridge that extends between the interventricular septum and some of the papillary muscles on the right side, and so it delivers the electrical signal, which we'll talk about in a future section, directly to the papillary muscles on the right side of the heart. The pectinate muscles are muscles found only in the atria, and you can't really see them very well in this image. You can see a cross section right here where some of them have been cut through. They have a comb-like appearance, and they are found on the anterior atrial walls. And then the muscle in the ventricles is called trabeculae carnii, and that's all of this muscle back here on the wall of the ventricle that is not these little papillary muscles that are sticking up attached to the chordae tendineae. So these are muscular ridges on the ventricle walls. So some differences between the right and left sides of the heart. So as we mentioned before, the right side is going to pump blood to the pulmonary circuit. The left side is going to pump blood to the systemic circuit. Both ventricles pump the same volume of blood every time they beat. However, the left ventricle has to use more force, so it has a much thicker muscular layer in the ventricle wall. So if you look at this image up here, you can see the muscle around the left ventricle is much thicker than the muscle around the right ventricle. When you're dissecting hearts in lab, you can tell if you're looking at the right or left ventricle by looking at the thickness of the muscle on the outside wall. The thickness on the left side is going to be much greater than the thickness on the right side. And you can also see in this view down here that shows when the ventricles are relaxed versus being contracted. You can see with how much more force the left ventricle contracts, and that is because even though they're pumping the same amount of blood, the left ventricle has to pump blood all the way out to the body, to your farthest capillaries, at the tips of your fingers and toes, whereas the right side only has to pump as far as the lungs, which aren't very far away from the heart at all, so it doesn't need as much force to pump that blood. Other noticeable differences between the right and left sides. The atrioventricular valve on the right side, also known as the tricuspid valve, has 3 flaps, while the atrioventricular valve on the left side, known as the bicuspid or mitral, has only 2 flaps. And only the right ventricle has a moderator band, as we discussed on the previous slide. In this section, we're going to look at the blood flow through the heart and the body, and this is something that you need to know for both lab and lecture. All right, so I'm starting with a blank slide here. So keep in mind that this is one big giant cycle, right? The blood is always going around in a circle, so there is no real technical start and ending point. So we just have to pick a place to start, and then we circle back to that place. And by tradition, we usually pick the right atrium as the starting place. Also, in my flow here, I'm going to use red to show places and structures that carry oxygenated blood, and blue to show places and structures that carry deoxygenated blood. And this is the same color code that is used in the images in your textbook and the models in lab. So again, it's one big giant cycle, so we just have to pick a place to start. So let's say we're going to start in the right atrium. So in the right atrium, we are carrying deoxygenated blood. It's going to pass through the tricuspid valve and into the right ventricle. So remember, the tricuspid valve is an AV valve between an atrium and a ventricle, and it's on the right side. From the right ventricle, it's going to pass through the pulmonary semilunar valve, which is at the base of the pulmonary trunk. The blood is then going to leave the heart through the pulmonary trunk. Shortly after it leaves the heart in the pulmonary trunk divides into right and left pulmonary arteries. This is one of the few arteries that actually carries deoxygenated blood in the body, so it's one of those exceptions of where you'll actually see a blue artery. And then from the pulmonary arteries, the blood is carried to the lungs. And notice that I switched colors here because oxygen is going to diffuse into the blood from the air in the lungs. So in the lungs, the blood becomes oxygenated. And this is why I switched to a red color. So blood is now going to leave the lungs carrying oxygen, and it's going to head back to the heart through the right and left pulmonary veins. Remember, arteries are going away from the heart, and then after you've gone through a capillary, you return to the heart through veins. So now we're in right and left pulmonary veins. And again, this is one of your exceptions. This is one of those very few veins that actually carries oxygenated blood. So the right and left pulmonary veins are going to carry this oxygenated blood into the left atrium of the heart. So now we're on the left side of the heart. The left atrium, the blood is going to pass through the bicuspid or mitral valve into the left ventricle. From the left ventricle, it is going to pass through the aortic semilunar valve at the base of the aorta, and then it's going to leave the heart through the aorta. And then from the aorta, it's going to branch into smaller and smaller arteries, which we will cover in chapter 21, eventually making its way out to all of the capillaries in the body. And at the level of the capillaries in your tissues, the oxygen is going to diffuse out of the blood and into the tissues so that your cells can use it. But that means that the blood is going to become deoxygenated. So the oxygen is leaving the blood and going out to the cells in the tissues. So notice I have switched back to blue. Now I am on my way back to the heart, so I'm going to be going through a series of veins, and eventually I'm going to get to the largest veins in the body, which are the superior and inferior vena cava. And from there, I'm going to return to the heart by going into the right atrium. So now I have done a full circle, and if I were a little blood drop, I would go back through the structures in this order. I would return back here, and then I would do it again. Right? So it's a cycle over and over again. You can imagine a little drop of blood just doing this little figure 8 over and over and over again. Right? So that's blood flow through the body and the heart. So again, notice that there is no movement between the left and right sides of the heart. When the heart leaves the right side of the heart, it has to go through this circuit to get to the lungs before it comes back to the heart. And then it has to go through the circuit where it goes to the body before it comes back to the heart. So also notice that now you're looking at this picture that's showing the blood flow. The tricuspid valve, it should now help you remember that that is preventing backflow into the right atrium. Because it's between the atrium and the ventricle, backflow would be going against these arrows. So this is preventing backflow into the right atrium. The bicuspid and mitral valve is preventing backflow back into the left atrium. The pulmonary semilunar valve is preventing backflow into the right ventricle. So again, remember, backflow would be the arrow going the opposite direction. And the aortic semilunar valve would prevent backflow into the left ventricle. So now that we have it drawn out like this, we can specifically say that over here on the left side, this is what we call the systemic circuit. The systemic circuit starts with the left atrium and ends with the superior and inferior vena cava. And over here on the right side, we have the pulmonary circuit, which starts with the right atrium and ends with the right and left pulmonary veins. So these are your two circuits. And again, the right side of the heart is pumping out to the pulmonary circuit, and the left side of the heart is pumping out to the systemic circuit. So hopefully that should enable you to understand the blood flow through the heart and the body. In this section, we're going to look at the fetal heart and differences between adult and fetal circulation. So the fetus is in a protected environment inside the uterus and getting all of the oxygen and nutrients it needs from the mother. So there is no air to breathe inside the womb, therefore there is no use for the lungs, therefore there is no use for the pulmonary circuit. So there are two structures in the fetal heart that lets the blood bypass the pulmonary circuit. The first one is the foramen ovale, which is an opening in the fetal interatrial septum, which allows blood to flow from the right atrium to the left atrium. This closes shortly after birth and becomes the fossa ovalis, which is a shallow depression in the interatrial septum of the adult. And then there is the ductus arteriosus, which is a small vessel that links the pulmonary trunk to the aorta in the fetus. And this also closes shortly after birth and becomes the ligamentum arteriosum, which is a fibrous band connecting these two great vessels. So here is a picture on the left side of the fetal heart, and the reason why it's shown in purple instead of nice red and blue is because oxygen circulation in the fetus is a bit different, because oxygen is not picked up in the lungs, but rather it's picked up in the placenta, and so that makes the oxygenation and deoxygenation of the different passageways and chambers a little bit different, so it's just represented in purple. But what you want to pay attention to is that the first structure we looked at, the foramen ovale, is right here. It's a hole between the right and the left atria. And then after birth, it closes up, and this is in a newborn's heart after delivery here on the right. So after birth, it will close up, and it will form a structure called the fossa ovalis. So in the adult heart, that would be the fossa ovalis. And then the other structure is the ductus arteriosus, which is shown right here. This is a little passageway between the pulmonary artery and the aorta. And again, shortly after birth, that's going to close up, and then that becomes the ligamentum arteriosum in the adult heart. So if we look at the fetal blood flow, we're not going to go into all of the detail like where the oxygen is picked up from the placenta. We'll talk more about that in chapter 21. But I wanted to show you these two structures that are in the fetal heart. And I've colored everything purple, again, because it gets a little bit more confusing when you try to follow oxygenated and deoxygenated blood in the fetus. So the two structures that we just learned about are there to bypass the pulmonary circuit because, again, if you're a baby in the uterus, you're not breathing air. You're getting all of the oxygen you need from the mother. So the lungs are not needed in the womb, so we do not need the pulmonary circuit. So both of those structures are there to bypass the pulmonary circuit. The first one of those structures, the foramen ovale, is between the right and left atrium, and it lets blood flow from the right to the left atrium again, and this is to bypass this pulmonary circuit. But not all of the blood will pass through this hole. Some of it is still going to go on to the tricuspid and to the right ventricle. So there is a second passageway between the pulmonary trunk and the aorta to again bypass the lungs and the pulmonary circuit. And the passageway between the pulmonary trunk and the aorta is the ductus arteriosus. So this allows blood flow from the pulmonary trunk to the aorta. So that is the fetal heart and an introduction to fetal circulation. In this section, we're going to talk about coronary circulation, which is blood flow to the heart muscle itself. So here's a picture of the heart, and we haven't talked about this before, but you should probably have already noticed that there are lots of blood vessels that are coming down to the heart muscle itself. So cardiac muscle cells work very hard. They are always working. They never get a break. They don't even get to sleep when you do. They are always working to pump the heart. And to do this, they need lots of oxygen and nutrients. Now you might think, well, the heart has all this blood going through it, so it should have all of the oxygen and nutrients that it needs, right? Well, the heart's own cells can't actually get oxygen and nutrients from the blood flowing through it because the walls are too thick, and this type of exchange only happens at capillaries. And so blood inside the chambers and inside the great vessels like the aorta and the pulmonary trunk, all of those have walls that are just way too thick for diffusion to happen through it. Therefore, the heart needs its own coronary circulation. So there is a left and right coronary artery shown here in the upper left picture that branch off from the base of the aorta. So these are the first two branches off of the aorta. So they're right here at the very base. There's the left coronary artery and there's the right coronary artery. So when blood is pumped into the aorta from the left ventricle, and then the left ventricle relaxes, that blood kind of falls backward a little bit from the reduced pressure, and that is called an elastic rebound. It's also why you have a aortic semilunar valve here to prevent that blood from falling back into the left ventricle. And when that blood falls back and the semilunar valve closes, it pushes the blood down into the coronary arteries. And so that is called the elastic rebound, and this is easier seen in a picture, so I actually found a video for you. So this video has no sound, so I'll be the narrator. So we're looking at the heart here, and it is showing you the coronary arteries, left and right coronary artery, and now we're going to zoom in and see where they come off of the base of the aorta. This is the aortic semilunar valve down here. So when blood pumps into the aorta, these arteries are actually blocked off by the flaps of the aortic semilunar valve. And then when it relaxes, if you notice, the blood flows back a little bit. That's the elastic rebound. So notice it pumps out and then it's going to flow back, pumps out and it flows back. And that's actually why we have those flaps there in the first place, is to prevent that flow back from going back into the left ventricle. But that elastic rebound also then helps to get that blood to go into the coronary arteries. And so here it is one more time. So you get a little bit of a swelling with the aorta when the blood pumps in, and then that elastic swelling is going to return to normal size, and that helps push the blood out into the coronary arteries. So the coronary arteries are then going to branch into smaller and smaller arteries. So you can see in this image, it's representing all the different little branches that these arteries are going to take. Eventually, you're going to get to the level of capillaries, and this is where the actual exchange takes place, which we're going to talk about in chapter 21. Then the blood is drained through cardiac veins. So because the cardiac cells are taking out the oxygen, these veins will be carrying deoxygenated blood. The cardiac veins are going to carry that deoxygenated blood, and they're going to dump it into the coronary sinus, which is visible on the posterior side of the heart. And it is right here in the coronary sulcus between the atria and the ventricles. So you can see the coronary sinus there. And then the coronary sinus is going to dump that blood back into the right atrium, where it joins back up with the normal circuit. And here is a picture where they have injected the blood vessels with latex to make them stand out more. So you can really see the thickness of the coronary sinus right here, and there it's actually going into the right atrium right there. So if we revisit our blood flow and we add to it the coronary circulation you can kind of think as just a little offshoot of the systemic circuit. So if you remember, with the systemic circuit, blood goes from the left atrium to through the bicuspid to the left ventricle, through the aortic semilunar into the aorta, then out to the body where the body cells take out the oxygen. Then it comes back through the superior inferior vena cava into the right atrium. Right, so that's the systemic circuit. The coronary circulation is like just a little offshoot of that. So from the aorta, blood is going to go into that left and right coronary arteries. It'll then go to the capillaries of the heart muscle. The heart muscle is going to take out the oxygen, so now we're switching over to deoxygenated blood. It's going to drain through the cardiac veins. The cardiac veins are going to dump the blood into the coronary sinus, and then the coronary sinus is going to go into the right atrium. So now we've just completed the systemic circuit. So coronary artery disease, or CAD, is when you have areas of partial or complete blockage of the coronary circulation. And usually this is going to happen in the coronary arteries because they are taking the blood to the heart muscle. So here is an image on the left. This would be a normal heart with an angiogram, which is where they can actually look at the health of the blood vessels of the heart. And on the right is a heart with advanced coronary artery disease, so you can see there is much less blood flow getting to the heart cells itself. Coronary artery disease is most commonly caused by atherosclerotic plaques, which are these fatty plaques that build up on the interior of vessel walls. And we're going to talk more specifically about them and what causes those in chapter 21 when we talk about blood vessels, but we're just introducing them to you here. So when you have reduced blood flow to an area of the heart, that area of the heart is getting less nutrients and less oxygen, and this condition is called ischemia. So whenever you have a reduction in oxygen or nutrients to an area, that's called ischemia, and because we're talking about the heart, this is cardiac ischemia. One of the first symptoms of coronary artery disease is angina pectoris, which a lot of people just call angina. And this is chest pain that is actually caused by a reduced blood flow to the heart. And it's usually a warning sign for an impending heart attack, which we're going to talk about next. So a myocardial infarction, or MI, is more commonly known as heart attack. And this occurs when part of the coronary circulation becomes completely blocked and the cardiac muscle cells start dying from a lack of oxygen. And remember, cardiac muscle cells do not divide, so if they die from a lack of oxygen, you are not getting them back. Even if you survive the heart attack, that part of your heart muscle wall is going to stay forever damaged. And so here is an image. So if you have a blocked coronary artery, all of the heart muscle downstream of that coronary artery is going to become damaged, and if those cells go without oxygen for long enough, they will die, and that is irreversible. Myocardial infarctions are usually caused by clots called thrombus that come loose from an atherosclerotic plaque. So what can you do to help avoid a heart attack or a myocardial infarction? Well, you can reduce your risk factors, so you can stop smoking, you can control high blood pressure if you have high blood pressure, lower your cholesterol intake because cholesterol contributes to those atherosclerotic plaques, you can maintain a healthy weight, and you can exercise. They do have a lot of medications that can help nowadays, like aspirin and a prescription medicine called Coumadin both reduce your blood's ability to clot. So by reducing the ability to coagulate or clot, you can reduce the risk of generating a thrombus. There are also drugs like nitroglycerin or nitric oxide, which cause vasodilation. So they cause the blood vessels to dilate or increase in their diameter. So that can help to overcome a blockage or a narrowed artery. The severity of a myocardial infarction is going to depend on which artery is blocked. So if it is a main artery, like one of those first two that come off at the base of the aorta, you're talking about most of the heart muscle being affected, which can lead to a very quick and sudden death. If it is a smaller artery, then you may only be cutting off blood flow to a smaller portion of the heart muscle, which would usually be survivable and just leave you with some potential problems in the future from having that area of your heart is now a dead area. Silent heart attacks, which are heart attacks that occur and have no pain, so people can be totally unaware of them, are the most dangerous because they could go unnoticed, untreated, and they could actually be a precursor to a fatal MI. And sometimes you never even know you might have had one of these until you have an EKG done, and the EKG is showing an altered signal from the heart muscle that died from that heart attack that you didn't even know you had. Women can also have different heart attack symptoms than men. So here is a picture from the American Heart Association of the most common heart attack symptoms. So in both men and women, you can get nausea or vomiting. You can get jaw, neck, or back pain. Men usually do always have squeezing chest pressure or pain. Women can have chest pain, but not always. So that's why a lot of women don't think it's a heart attack, because their chest doesn't hurt. Shortness of breath is also in both sexes. Women will oftentimes feel pain or pressure in their lower chest or up their abdomen, so they might think it's something wrong with their intestines, and again, they don't think it's a heart attack. Women are also more likely to experience fainting, indigestion, or just extreme fatigue. So if you do have a heart attack, you can get balloon angioplasty. Well, this is even if you have a blocked artery and haven't yet had a heart attack. And with balloon angioplasty, they insert a catheter with a balloon at the tip to open up the narrowed artery that has the atherosclerotic plaque on it. And usually accompanying this procedure, they'll insert a stent, which is a wire mesh cage that is inserted to hold the vessel open. So in this image here, this is the catheter going in, and then the balloon is expanded to push that vessel open and to reduce the size of that atherosclerotic plaque. And again, usually as they inflate the balloon, they're also putting in a stent, and a stent is this little wire mesh cage that's going to stay in there after they withdraw the catheter and hold the vessel open. You can also have a coronary artery bypass, in which they take peripheral arteries or veins, usually from your legs, and they graft them in to bypass the clotted area. So they would like attach a artery that they took from your leg, and they would attach it up here and then connect it down here. So it would actually bypass the occluded portion or the clotted portion. And there are 4 different coronary arteries that you can do this surgery on, and so that's why you hear about a single, double, triple, or quadruple coronary artery bypass. So before we get really into the cardiophysiology, I want to introduce you to the 3 main types of cardiac cells that we're going to talk about, and these are the pacemaker cells, the conducting cells, and the contractile cells. So cardiac muscle tissue can contract on its own, and this is a property called autorhythmicity, the ability to contract without neural or hormonal stimulation. So they do not need a signal from either the nervous system or the endocrine system. You can put some cardiac muscle cells in a dish and they can beat on their own. So the nervous system and the endocrine system can still adjust the heart rate and the force of contraction, but they are actually not needed for the heart to beat. And I actually have a video to show this to you. Okay, so I'm going to play this video, and what we're looking at here are 2 separate heart cells in a petri dish, and they have joined them together. They've put a little channel here so the 2 heart cells can talk to each other, and you can see this one up here is beating, this one here kind of appears to be listening, and then after a few minutes, right there, you see they start contracting in sync. So they start contracting in unison. So now they're both contracting faster. So they very quickly link up. And here we have another quick example. This is with 4 different cardiac cells. So in the beginning, it looks like the 2 left are beating together and the 2 right ones are beating together. And then again, they have little channels so that they can communicate, and it doesn't take long until all 4 of them start beating in unison. So this is the principle of autorhythmicity. So they not only can beat by themselves, but they can join up with adjacent cardiac cells and time their beat with the ones of the cardiac cells next to them. So that was the principle of autorhythmicity. So now let's talk about the 3 main types of cardiac muscle cells. There are pacemaker cells, which are the cells that can initiate the contraction signal. There are conducting cells, which are the cells that conduct the contraction signal around to the different parts of the heart. And then there are the contractile cells, which are the cells that do the actual contracting. And this is the largest population that makes up 99% of the cardiac muscle cells. So let's first talk about the pacemaker cells. These are cells that can initiate the electrical impulse that causes a heartbeat. And so these cells do not have a stable resting membrane potential. We talked about resting membrane potentials last semester in A&P 1. And we talked that in most cells, it's pretty stable and something has to happen to change it. But with the pacemaker cells, as soon as they hit resting, so as soon as they finish an action potential, they immediately start depolarizing to a new action potential. And this is what it looks like. So they have an action potential, they depolarize, they repolarize, they return to resting, but then they immediately start depolarizing again to a new action potential. And this is called a pacemaker potential. And it means they don't need to be stimulated in order to fire an action potential. They do it automatically. And pacemaker cells are found in the sinoatrial node, also called the SA node, and the atrioventricular node, also called the AV node. So the SA node is located in the posterior wall of the right atrium, and the AV node is located in the junction between the atria and ventricles near the coronary sinus. So the pacemaker cells set the normal heart rate and the normal heart rhythm. So when we talk about heart rhythm, we're talking about the order in which the different chambers of the heart beat. Like it starts with the atria and then it moves to the ventricles, and we'll look at that in a moment when we look at the EKG or ECG. And so a term you should be familiar with, sinus rhythm, is a description of a normal heart rhythm. So the rhythm is the order in which the structures of the heart beat. Heart rate is how many times it beats per minute. So the SA node can fire off 60 to 100 action potentials per minute, and the AV node cells, pacemaker cells, can fire off 40 to 60 action potentials per minute. But since the SA node is faster, it actually sets the normal heart rate, and it ends up causing the AV node to fire sooner than it otherwise would. So as long as the heart is completely normal and there's no damage to it, the SA node will be the one that is driving the heart rate. If something happens to the SA node, the AV node can then take over and become the primary pacemaker. But in a normal heart, the primary pacemaker is the SA node. The AV node has a limit. It cannot fire more than 230 action potentials per minute. So that is the limit on heart rate. So without actually taking specific drugs to change the membrane potentials, the heart cannot beat faster than 230 beats per minute. And that's okay, because once heart rate gets above 180 beats per minute, it becomes dangerous anyway for reasons that we'll talk about later on when we talk about regulation of heart rate. So that is the pacemaker cells. Note that the resting heart rate is normally lower than the SA node rate due to neural innervation from the parasympathetic nervous system. This is your rest and digest nervous division. And if you remember from last semester, the parasympathetic nervous system lowers your heart rate. So when you're sitting around doing nothing, you actually have active input from the parasympathetic nervous system that is slowing the rate at which the SA node is firing. So the conducting cells are the cells in the heart that conduct from the, or they conduct that electrical signal from either the SA node to the AV node, and then from the AV node out into the different areas of the myocardium, which is the muscle of the heart. So the conducting cells can connect both the SA node to the AV node and also distribute the electrical signal throughout the myocardium. Most conducting cells are smaller than the contractile cells that do the actual contracting, and they have fewer myofibrils, which is where you find the sarcomeres and the thick and thin filaments. The exception is a set of conducting cells called the Purkinje cells, which are located in the ventricles. These are actually larger than contractile cells. If you have any damage to your conducting cells, this can alter the normal heart rhythm, and we'll see that when we look at the section on ECGs. And for example, if you have a myocardial infarction or a heart attack, we talked about how that could lead to death of cells in the heart. And if some of those cells include the conducting cells, the conduction signal no longer travels along the same path that it normally should, and that will show up as an altered rhythm on an ECG, what used to also be called an EKG. And that leaves us with the contractile cells. The contractile cells are the bulk of the myocardium. They're 99% of the cardiac muscle cells, and these are the cells that cause the heart to contract. So these guys are joined together by intercalated discs, as we introduced you to last semester in Chapter 4. And an intercalated disc is a connection made up of both desmosomes and gap junctions. So you have gap junctions directly between one cardiac muscle cell and the neighboring one. You've also got these desmosomes, which are these really strong connections between each cardiac muscle cell. So the desmosomes hold the cells together tightly so they don't separate during the force of a contraction. And the gap junctions allow the electrical signal to flow directly from cell to cell, so there is no need for synapses, which result in synaptic delays, and so the signal can be immediately transferred from one cell to the neighboring cell. So other than that, the cardiac muscle cells have a lot in common with the skeletal muscle cells you learned about last semester. They have the sarcomeres made up of the thin and thick filaments, with the actin and myosin proteins that interact with each other to cause the shortening of the sarcomere, and all of that works to make them contract, just like you learned with skeletal muscle in Chapter 10. And then we have a chart here to compare the cardiac contractile cells with the skeletal muscle cells, also known as fibers, that you learned about last semester. So this should just be a review for you. So if you remember, in terms of the nucleus, cardiac contractile cells typically only have one nucleus. Skeletal muscle cells can have multiple nuclei up to hundreds. Cardiac cells are going to have abundant mitochondria, much more abundant than those of the skeletal muscle cells, because your cardiac cells have to stay supplied with ATP because they have no other choice but to keep beating, or else the heart will stop working and then the whole body will stop working. For the same reason, cardiac muscle cells have a very extensive blood supply, much more extensive than the blood supply to skeletal muscles. In terms of reserves, cardiac muscle cells have a huge reserve of myoglobin, which is a protein very similar to hemoglobin, but myoglobin is used to store oxygen. You also have a nice big reserve of lipids and glycogen. And compared to skeletal muscle fibers, there is very little myoglobin, very little lipids, but they do have extensive glycogen in your skeletal muscles. The stimulus for contraction, as we already mentioned with the cardiac cells, you have autorhythmic pacemaker cells that can allow the heart to beat on its own, whereas skeletal muscles don't contract unless you have input from a somatic motor neuron. The intercellular connections, cardiac muscles are directly connected to each other through intercalated discs. They can also be connected with connective tissue fibers. In skeletal muscles, you only have the connective tissue fibers, no intercalated discs. The resting state, cardiac muscle cells never rest. If they rested, your heart would stop beating and you wouldn't live for long. In skeletal muscles, at rest, they use aerobic respiration, so they're using their mitochondria to make ATP, and they're using up oxygen. The active state of cardiac muscle cells is going to be the aerobic respiration, where they're using their mitochondria and using up oxygen. Skeletal muscles have the option to do anaerobic respiration, as we talked about last semester. Cardiac muscle cells do not have that option. And then cardiac muscle cells, we're going to look at the actual action potential that takes place in these muscle cells, and they have what's called a long refractory period, and this prevents them from undergoing those tetanic contractions. And if you remember from last semester, skeletal muscles do have that possibility for tetanus. So if you stimulate a muscle cell at a very high frequency stimulation, you can get that maximum tetanic stimulation where you get that plateau of really high force, but it's like the muscle is stuck there in a contracted state. And you don't want that in a cardiac muscle cell, because if you had a tetanic contraction, that would essentially mean that your heart contracted and then didn't relax. And if it can't relax, it can't beat. So the heart has to be able to contract, relax, contract, relax, contract, relax. So the heart muscle has to be unable to undergo tetanic contractions, because if it could, that would be very dangerous. So now that we know about the different types of cardiac cells, in this section we'll walk through the actual conduction system of the heart. So we have a picture here, and before we start walking through this system, I want to point out some terminology to you. Your textbook doesn't introduce this into a later section, but I think it's good to go ahead and know this now. When you see the word systole, that refers to the contraction of a chamber, and diastole refers to relaxation of a chamber. All right, so let's look at the steps of the conduction system. So number one, the sinoatrial or SA node is going to generate an action potential. So remember, in a normal, healthy heart, the SA node is going to be the pacemaker. The action potential is then going to travel through these internodal pathways, and as the signal passes through these internodal pathways, that is going to be a signal for the atria to contract, both the right and left atria at the same time, and that would be called atrial systole. Then the signal arrives at the AV node, or the atrioventricular node. It slows a little bit at this point because of the anatomy of the cells in this area, and the slowing of the signal here allows the atria to finish contracting, which is going to allow the ventricles to finish filling up with blood. The signal is then going to pass through this little spot right here, which is called the atrioventricular or AV bundle. This is also still commonly called the bundle of His. So in other textbooks or websites, you may see it called the bundle of His instead. The signal is then going to move out through the left and right bundle branches. So the AV bundle splits into a left and right bundle branch. Then the signals are going to finally go out into the ventricles through the Purkinje fibers, again on both the right and left sides. And the signal also is going to pass through that moderator band on the right side. So we talked earlier about the moderator band in the right ventricle functioning as a path for the conduction signal. The Purkinje fibers are then going to distribute the signal to the ventricular myocardium, aka to the contractile cells in the ventricle, and this is going to be the signal for the ventricles to contract. And we would call this ventricular systole. So note that the papillary muscles in the ventricle get the signal first so that they can start contracting to pull on the chordae tendineae, which helps to stabilize the AV valves right before the ventricles contract. So I have a little video to show you. It's very quick. Forgive the roughness of the video. This used to be a flash animation, and you can't get it anymore because modern web browsers don't support flash animations anymore, but someone was able to grab it before it disappeared forever.
Action potentials originate in the sinoatrial node and travel across the wall of the atrium from the sinoatrial node to the atrioventricular node. Action potentials pass slowly through the atrioventricular node to give the atria time to contract. They then pass rapidly along the atrioventricular bundle, which extends from the atrioventricular node through the fibrous skeleton into the interventricular septum. The atrioventricular bundle divides into right and left bundle branches, and action potentials descend rapidly to the apex of each ventricle along the bundle branches. Action potentials are carried by the Purkinje fibers from the bundle branches to the ventricular walls. The rapid conduction from the atrioventricular bundle to the ends of the Purkinje fibers allows the ventricular muscle cells to contract in unison, providing a strong contraction.
Okay, so some terminology related to this section. Bradycardia is a slower than normal heart rate. Tachycardia is a faster than normal heart rate. And there is also something called an ectopic pacemaker. So the word ectopic just means out of place, like an ectopic pregnancy is a pregnancy that is not occurring in the correct place, which would be inside the uterus. So an ectopic pacemaker is when an abnormal conducting cell or even a contractile cell starts generating an action potential at a higher rate than the SA node or the AV node, and it causes disruptions in that normal conduction pathway that we just looked at. So it disrupts the timing of the normal cardiac rhythm. This happens to everybody from time to time, and so acutely this is what can just be felt as like a palpitation when it feels like your heart does a funny little flip-flop in your chest. That's when it just had an irregular heartbeat and the signal didn't quite pass in the correct order through all of the structures. However, if it happens chronically, it can lead to bradycardia, tachycardia, or even what we call ventricular fibrillation, which is VFib. And ventricular fibrillation is when the ventricle is just quivering instead of actually contracting, and so it's not pumping blood at this point. So you see this a lot in medical dramas where someone comes in to the ER and someone shouts, "He's in VFib," and then they grab the paddles and they shock him. And then you might hear, "Oh, we've reestablished the normal sinus rhythm." And so now you should know what all of that means. In this section, we're going to talk about the electrocardiogram, or ECG. And notice I occasionally say EKG without realizing it. An EKG came from the original German spelling of electrocardiogram. So these two terms are still used interchangeably today. So an electrocardiogram, an ECG, or an EKG looks like this. So it's a series of waves, intervals, and segments. So the electrocardiogram allows us to see and measure the pathway of the conducting system that we just talked about. Waves are large changes from the baseline, and there are three main waves. There is the P wave shown here, then you have the very large QRS complex, and then the T wave is shown afterwards. So this is actually, this graph here is showing you two back-to-back heartbeats. This would be the first heartbeat, this would be the second heartbeat. Segments are periods from the end of one wave to the start of the other. So for example, the PR segment shown here would be from the end of the P wave to the start of the QRS complex. ST segment would be from the end of the QRS complex to the start of the T wave, and etc. And intervals are combinations of segments and waves. So like the ST interval here includes this segment, which is the ST segment, and the T wave. Now, in lecture, I am not going to test you on the names of the different segments and intervals, but you will have to learn those for lab, and I am posting my lab interactive online lesson that you can use to review that information. However, for lecture, I do want you to know the three waves and what they represent. And we'll cover that information on the upcoming slides. So what the ECG is actually showing, as I mentioned before, is the pathway of the electrical signal through the conduction system. So we're going to look at an ECG tracing on the right side of your screen, and we're going to compare it to what's actually going on in the heart on the left side of your screen. So remember that the SA node is where you have those pacemaker cells that are going to start the electrical signal, but at this point there is no current signal going on in the heart, and so the line would be at baseline on the ECG. Next, the SA node will actually fire its action potential, and that action potential will travel through those internodal pathways that go through both atria. So at this point, as that signal passes through the atria on the way to the AV node, the atria are getting the signal to contract. So on the ECG tracing, this is where you'll see the little depolarization called the P wave, and this is representing the atria are contracting, and you can also say it's atrial depolarization because the signal inside the heart is becoming more positive, which is a depolarization. So now the signal has reached the AV node, where it's going to slow down a little bit to let the atria finish contracting. And so at this point, there is no actual current signal that's being measured because the signal has slowed down. And so we finished the atria contracting, shown as the P wave, and now we are back to the baseline for a brief period of time. And just FYI, this would be called the PR interval. So then the signal is going to travel through the AV bundle, and the bundle branches and out through the moderator band in the right ventricle. So at this point, we are starting to see the ventricles are getting ready to contract. So you see this small change right here represented by the Q, but we're not going to look at the Q separately. We're going to look at the whole QRS complex. But just at this point, just know that the signal is moving down through the interventricular septum and getting ready to go out into the ventricles. So now the signal is going through those Purkinje fibers into the ventricles. The ventricles are getting the signal to actually contract, and that's when you're going to see this very large QRS complex, also called ventricular depolarization, and this is the point at which the ventricles are actively contracting. Note that the atria are relaxing at this point, but we don't see this signal, mostly because it's masked or hidden by this larger QRS complex. So the electrical activity taking place in the ventricle is overriding whatever electrical activity we could see going on in the atria. And then your textbook doesn't add a number 6, but I did so that we could continue the discussion of the waves. So after the impulse has gone through the Purkinje fibers and through the myocardium of the ventricles, it's going to die out, and the ventricles are then going to relax, and this is called repolarization. And we'll see this on the ECG as the T wave. So the T wave is the repolarization of the ventricles, and that is when they are relaxing. So that is what the P, QRS, and T waves represent. And so all of those segments and intervals that you're going to talk about in lab are just the time periods for the signal to travel, say, through this structure or from this part of the conduction system to another part of the conduction system. And then when you look at an ECG, right, each of these is a separate heartbeat. So one heartbeat will have a P wave, a QRS complex, and a T wave. So this is one heartbeat here. Then this is the next heartbeat, this is the next heartbeat, and this is the next heartbeat. So the whole cycle is just going to keep repeating as the SA node keeps sending signals at regular intervals through the conduction system. So a cardiac arrhythmia is when you have an abnormal pattern of the cardiac electrical activity. And your textbook gives you several examples, and I'm not going to test you on these examples or being able to recognize what the different arrhythmias look like. You'll get that if you go on to nursing school or another type of healthcare field type of school. You'll go into that level of detail, but we're not going to do that just quite yet. I did, however, want you to know what fibrillation means, because we've already mentioned that. Fibrillation is when you have a quivering of a chamber instead of a contraction. So over here on the right side, you can see atrial fibrillation is when this is happening inside the atria, and then the more severe ventricular fibrillation, also called VFib, when it's happening inside the ventricles. And so I blew up this one here. Ventricular fibrillation is rapidly fatal because the ventricles are quivering and they stop pumping the blood through the body. So cardiac arrest is when the heart suddenly and unexpectedly stops beating. And this is often due to an arrhythmia, particularly the ventricular fibrillation. This is one of the most common causes of cardiac arrest. And this is why the little paddles that you use to shock someone's heart is called a defibrillator, right? It's usually because the heart is in fibrillation and you're trying to defibrillate it and get it back to a normal or sinus rhythm. So by putting an electrical shock into the heart, the hope is that you reset the conduction system and it goes back to a normal rhythm, also called a sinus rhythm. In this section, we're going to talk a little bit more about the cardiac contractile cells, which make up 99% of the cells in the heart. And so we will cover the cardiac action potential and the importance of calcium. So action potentials were covered in great detail back in chapter 12. So I am only going to cover the highlights here and what is different from what we learned before. You can go back and refer to chapter 12 if you need a refresher on what an action potential is. So when we did talk about action potentials, we talked about them in neurons, and we talked a lot about how neurons had a resting membrane potential of negative 70 millivolts. Well, cardiac contractile cells in the ventricle actually have a resting membrane potential that's even more negative. So the resting membrane potential of these cells is negative 90 millivolts. And the cardiac action potential has three main phases. First, it has a rapid depolarization phase, and this is very similar to what we saw in the neurons. You get the opening of voltage-gated sodium channels, and you get sodium that rushes into the cardiac cell, which makes the membrane potential very rapidly move in the positive direction. This is new. This is called the plateau phase, and in this phase, you get the opening of voltage-gated calcium channels, which cause extracellular calcium to come into the cardiac cell. And because we're, again, adding positive charges to the inside of the cell, we are keeping it depolarized for a relatively long period. And so this is called the plateau phase, and it is due to calcium coming into the cell. Then we have the repolarization phase that is similar to what we saw in neurons, where we get the opening of potassium channels. This causes potassium to leave the cell, which is going to return the membrane potential back down to resting. In cardiac action potentials, we don't have that hyperpolarization phase that we have in neurons. And that is because the resting membrane potential is already pretty much at the equilibrium potential for potassium. So the calcium-mediated plateau phase, that is the most different from what we learned before, actually gives the cardiac muscle a long refractory period. And to refresh your memory, this was also covered in chapter 12 in A&P 1. The absolute refractory period is when the membrane cannot respond to another stimulus, no matter how strong it is. And the relative refractory period is when the membrane can respond to another stimulus, but that stimulus must be larger than normal. So if we look at the refractory period of a cardiac muscle cell compared to a skeletal muscle cell, you can see the difference. So at the top here in both the top and bottom images, so up here these two graphs are showing a skeletal muscle fiber. The two graphs at the bottom are showing a cardiac contractile cell. And so in the skeletal muscle fiber, the blue represents the absolute refractory period, and the salmon colored represents the relative refractory period. So you can see how short both are. And when you get this action potential, it's followed by a very fast contraction. Whereas in the cardiac muscle cell, you have a very long absolute refractory period. Even the relative refractory period is longer than it would be in a skeletal muscle fiber. And so when you get contraction of a cardiac cell, it is a more drawn out event, and it is not so quick and fast as a skeletal muscle cell. And this is actually why cardiac muscle cells cannot go into tetany, which is that stage of tetanic contractions where you have a high frequency stimulation that causes the muscle to essentially contract and stay contracted. And here is an example of what tetany would look like if it was in your arm. And it's a good thing that our cardiac muscle cells can't do this, because if they basically contract and then don't relax, they can't pump blood. So in order for the heart to pump blood, it has to constantly contract and relax, then contract and relax. So it has to have a mechanism to prevent it from going into like a spasm where it is constantly contracting. So that leads us to the importance of extracellular calcium. And if you haven't figured out by now, blood calcium levels are very important. So we started talking about blood calcium levels way back in chapter 6, and it doesn't seem like we've stopped talking about them since. So hopefully that drives home the point of just how important blood calcium levels are. So cardiac tissue is highly sensitive to changes in fluid calcium concentrations. So by fluid, I mean both the blood calcium levels and the calcium levels in the interstitial fluid. So 20% of the calcium that is needed for a cardiac action potential actually comes from the extracellular fluid. This is very different than the way skeletal muscles work, because if you remember from chapter 10, skeletal muscles store all the calcium they need in the sarcoplasmic reticulum inside the cell, also called the SR. And while cardiac muscle cells also have an SR full of calcium, they also need calcium to come into the cell from the extracellular fluid. So that's different. So the calcium that comes in from the extracellular fluid is actually required for cardiac muscle cells to release the calcium that they have stored in their sarcoplasmic reticulum. So in the muscle cells, the skeletal muscle cells we learned about last semester, the action potential itself was enough to release the calcium from the SR. It's a different mechanism in cardiac muscle cells. Calcium has to come in from outside of the cell before the cell can release the calcium that is in the sarcoplasmic reticulum. So there's a lot going on in this picture, so I don't want you to get too overwhelmed. What I want to point out here, this little red compartment, this is the sarcoplasmic reticulum in a cardiac cell, also known as a myocyte. And so it does have calcium inside of it, but you have to have calcium come into the cell through calcium channels in order to release the calcium from the SR. And then the calcium is, again, very important to start that contraction cycle, like we talked about last semester in chapter 10, where it binds to the troponin, which causes the tropomyosin to move out of the way of the active sites of actin. And so then you can get interactions between actin and myosin, and that causes the sarcomeres to shorten, which lets the muscle contract. All of that's still the same. The difference is that cardiac muscle cells need calcium to come in to the cell from the outside before they can release the calcium in their SR. So both hypercalcemia and hypocalcemia will affect the heart. Hypercalcemia is a blood calcium level that's too high, and hypocalcemia would be a blood calcium level that is too low. Both of those will negatively and adversely affect heart function. Cardiac muscle cells also need a lot of energy, and I mentioned this briefly before, but it's worth it to repeat it. Myoglobin is a protein used inside of cells to store extra oxygen. It's related to hemoglobin, but it's a slightly different protein. So it binds to oxygen very similar, in a way very similar to the way that hemoglobin does. Cardiac cells have an extensive myoglobin reserve so that they can keep oxygen available inside their cells, so their mitochondria never run out, and so that they can make sufficient ATP for their contractions, because you don't want your cardiac muscles to ever reach a fatigue level where they run out of ATP and can no longer contract, because your heart always needs to beat. So now we will walk through the stages of the cardiac cycle, which are the events that take place from one heartbeat to the next. So the cardiac cycle are the events between the start of one heartbeat and the start of the next. And you can see the whole cycle right here in this image. So the cycle is going to consist of a series of contractions and relaxations. So you'll have atrial systole, which is contraction of the atria, followed by atrial diastole, which is relaxation of the atria. You'll also have ventricular systole and ventricular diastole. So as a reminder, systole is the contraction of a chamber, and that's when you'll have the highest pressure inside that chamber. And diastole is the relaxation of a chamber, and that's when you'll have the lowest pressure inside that chamber. And these cycles of contraction and relaxation is why blood pressure is always reported with two numbers. So like our normal blood pressure is called 120/80. And that first number is called the systolic pressure. So this is the pressure during ventricular systole, or when the ventricles are contracting. And the second number is the diastolic pressure, which is the pressure when the ventricles are relaxing. And we're going to go into more detail with blood pressure when we get to chapter 21, because that's more of a measurement of the force or pressure of the blood in the arteries. So one cardiac cycle is going to be composed of an atrial systole followed by an atrial diastole, and then ventricular systole, which can be divided into two phases, followed by ventricular diastole, which can also be divided into two phases. So that would make up one cardiac cycle. So before we get started, some terminology to keep in mind. So if you see diastole or systole and you don't see the word atria or ventricle in front of it, then it is assumed to refer to the ventricles. So like earlier, I told you that blood pressure, that first number is systolic pressure and the second number is diastolic pressure. Well, since it doesn't specify whether it's the atria or the ventricles, you're to assume it's the ventricles. So this is the pressure when it's systole in the ventricles and pressure when it's diastole in the ventricles. Also some terminology that we're going to introduce you to in this section, and then we're going to go into more detail in the subsequent section. The first one is end diastolic volume. Remember, diastolic or diastole is relaxation. So this is the volume in the ventricles at the end of their relaxation. Again, it doesn't specify end ventricular diastolic volume. So if it doesn't say atria or ventricles, you're to assume it's talking about the ventricles. End systolic volume would be the volume in the ventricles at the end of their contraction. Again, remembering that systole is contraction. And then the stroke volume is the amount of blood pumped out of a ventricle with one contraction. Also keep in mind that as the heart rate increases, the time spent in diastole, which is the relaxation phase, is reduced more than the time spent in systole, which is the contraction phase. So the faster your heart beats, the less time you have that relaxation period where the chambers fill with blood. So as heart rate increases, you're actually pumping less blood because it has less time to fill up. And this is why heart rates over 180 beats per minute become dangerous, because while the heart can beat that fast, at some point you actually stop pumping blood, so you lose the efficiency of being able to fill up with blood before contracting again, and so you end up actually pumping out very small volumes of blood. All right, so let's look at the cardiac cycle, and we're going to start with atrial systole. So again, this is a cycle, so you just kind of have to pick a place to start. So we're going to kind of work from the top down. So we're going to pick atrial systole as our starting place. And note that when atrial systole starts, the ventricles are already 70% full of blood. And you'll see why by the time we get to the end of the cardiac cycle. But for now, just take my word for it that at the start of atrial systole, the ventricles are already 70% filled with blood. So with the atrial systole, the atria contract, and that is going to squeeze a little bit more blood into the ventricles. So it's going to top off the ventricular volume. It's going to add that remaining 30% to make the ventricles full. Notice that the AV valves are open. This would be the tricuspid and bicuspid or mitral are open so that blood can move from the atria to the ventricles. At this point, after the atria contract and tops off that volume, the ventricles now hold the maximum amount of volume that they're going to hold for this cycle, and that is called the end diastolic volume, or EDV. So the next phase would be atrial diastole, and this happens at the same time that your ventricular systole phase 1 starts. So the atria are now fully relaxed. They are no longer contracting. That is why it's atrial diastole. However, you have pressure start to build up in the ventricles, so the muscle is starting to contract. And this increasing pressure As the ventricles are getting smaller because of the muscle contraction, the increasing pressure is going to cause the AV valves to close. So it's going to snap shut the tricuspid and bicuspid. And now notice that at this point, all the valves are closed. The AV valves are closed and the semilunar valves are closed. Contraction in the ventricles at this stage is called isovolumetric contraction because there is tension building up in the muscle of the ventricles, but no blood is flowing anywhere yet because all of the valves are still closed. Then we move into the ventricular systole phase 2. And at this point, the pressure has built up enough inside the ventricles that it forces the semilunar valves open. So now at this point, the semilunar valves are open, the AV valves are still closed. So now blood is ejected from the ventricles out into the aorta and the pulmonary trunk, and this is called ventricular ejection. So the amount that is actually ejected with this contraction of the ventricles is called the stroke volume, and the amount that is left over when the contraction is finished is called the end systolic volume, or ESV. Then we move into ventricular diastole phase 1. So the semilunar valves are going to close because the pressure is dropping inside the ventricles, and that causes these valves to close back. You actually have a bit of force of the blood in the vessels going back against the semilunar valves too. Remember, that's that elastic rebound that helps the blood move out into the coronary arteries that we talked about earlier. Notice that at this point, all valves are now closed. Semilunar valves are closed, AV valves are closed. And meanwhile, while this is happening, the pressure inside the atria has dropped enough that blood is just passively flowing from the veins, so the vena cava and the pulmonary veins, into the atria. So the atria are now filling up with blood. Relaxation in the ventricles at this stage is called isovolumetric relaxation because relaxation is occurring, but no blood is yet flowing anywhere into or out of the ventricles. So that is why it's called isovolumetric relaxation. So when you see isovolumetric, that is referring to a period when all of the valves are closed, so no blood is moving into or out of the ventricles. So then we have ventricular diastole phase 2. And in this phase, the pressure inside the ventricles from the relaxation has dropped enough so that the AV valves snap open. So now you've opened the tricuspid and the bicuspid, and as soon as those valves open, passive movement of blood starts to move blood from the atria into the ventricles, and blood is still coming into the atria from the different veins. And all of this is what we call passive movement of blood because there's no muscle contraction going on right now. This is all blood moving to areas of low pressure. So blood is not being pumped anywhere at this point. It's passively moving from an area of high pressure to low pressure. So just like we talked about diffusion in A&P1 where solutes move from a high to low concentration, fluids always want to move from an area of high pressure to low pressure. So the decreased pressure in the ventricles from the relaxed muscle is going to cause blood to passively flow into both the atria and the ventricles. This passive flow of blood into the ventricles is going to fill the ventricles up to their 70% of their full volume. So this will fill the ventricles up 70% full of blood. And then that brings us back to the beginning, where we started with atrial systole. And remember, I told you at the beginning, the ventricles are already 70% filled, and that was because of that passive blood flow that happens at the end of ventricular diastole. And then at the start of the next cycle, the atria contracts, which tops off that ventricular volume. That adds the remaining 30%. The AV valves are still open, the semilunar valves are still closed, and then when it's done with this, you'll have that end diastolic volume. So note that, again, the passive movement of blood into the ventricles fills them up to 70% full. When the atria contract, it is only adding the remaining 30% of the volume. So atrial systole only contributes to 30% of the ventricle volume. So this is why you can have severe damage to the atria, like for example with a heart attack or some other injury, and you can still survive because you can still survive off that 70% of the blood that fills up the ventricles passively. You don't necessarily need that extra 30% contributed by the atria. So the pumping of the atria actually contributes very minorly to overall blood flow. Ventricle contraction, on the other hand, is extremely important. Ventricle contraction is how we get blood out into the arteries. So heart failure is caused by damage to the ventricles. When the ventricles can't pump enough blood to get it out to the peripheral tissues and organs, that is the definition of heart failure. So heart failure would be an insufficiency of the ventricles. And then the last thing we need to talk about is cardiodynamics, and we're going to start with cardiac output. So there's an equation to know: cardiac output equals stroke volume times heart rate. So the cardiac output is the amount of blood pumped by the left ventricle in 1 minute. Both ventricles pump out equal amounts of blood, but by tradition, cardiac output is defined as the amount pumped by the left ventricle. So the cardiac output equation is shown here at the bottom. Cardiac output, or CO, equals heart rate times stroke volume. So the heart rate is the number of heartbeats per minute, and the stroke volume is how much blood is pumped out of a ventricle with one contraction or one beat. So if you think about it, the equation makes sense. Cardiac output is how much blood is pumped in a minute. So to get that, you need to know how many beats per minute and how much blood is pumped out with each beat. So you take the beats per minute, multiply that by the blood pumped out with each beat, and that will tell you the total amount of blood pumped out in 1 minute. So stroke volume also has its own formula you need to know. Stroke volume is calculated by taking the end diastolic volume, which is the amount of blood in the ventricles when they are full at the end of their relaxation period, and minus the end systolic volume, which is how much blood is left in the ventricle after it squeezes or contracts, and that gives you the stroke volume, or how much was pumped out. And so the next couple of sections, we're going to go over the factors that affect both of these components of cardiac output. But first, a little bit more about the stroke volume. So again, stroke volume is the amount of blood pumped out of a ventricle with a single contraction, so in one heartbeat. And we can calculate stroke volume by taking the end diastolic volume and subtracting the end systolic volume. So your textbook gives you an example of like using a pump. So if the pump is full, so after you've added water to the pump, and you have maximum amount of water in here, that would be the end diastolic volume. And then if you squeeze some out, what is left when you're done squeezing would be the end systolic volume. So the stroke volume is going to be the difference between those two, or how much you actually squeezed out. I have another analogy that I like to use. I think it makes it a little bit easier, in my opinion anyway. Think of the end diastolic volume, or EDV, as being your starting volume right after you have finished relaxing and filling the ventricles. And so imagine this being like a tube of toothpaste before you have squeezed it, right? So it's full of toothpaste. So that would be your end diastolic volume. The end systolic volume, or ESV, is how much blood is left inside the ventricle after contraction. So my analogy is, think of after you've squeezed that toothpaste tube as hard as you can, there's always going to be a little bit left in the tube. And so the end systolic volume is what is left over after you are done squeezing. So that's how much toothpaste is in the tube after squeezing. So if you take how much was in there before you squeezed, and you subtract how much is left after you squeeze, then it should stand to figure that the amount you squeezed out would be the stroke volume. So the stroke volume is how much blood is pumped out with one contraction, and in this analogy, this would be how much toothpaste you squeezed out when you squeezed the tube. So in this section, we're going to look at factors affecting the heart rate. And again, the idea is overall we're trying to see what can affect cardiac output. And remember, cardiac output is stroke volume times heart rate. So in this section, we're going to look at what can affect the heart rate. So one of the biggest factors on heart rate is the autonomic nervous system, which we covered in A&P 1. So as a reminder, the sympathetic division, that's your fight or flight division, increases heart rate. And the parasympathetic division, which is your rest and digest division, decreases your heart rate. And in fact, the parasympathetic division controls your resting heart rate. If you remember from earlier, the pacemaker cells in the SA node can depolarize automatically up to 100 beats per minute. And so the parasympathetic division is actually constantly giving them a stimulus to tell them to slow down. And that is why the normal resting heart rate is usually between 70 and 80 beats per minute. And we know that if you damage the vagus nerve, and the vagus nerve carries parasympathetic signals from the brain to the heart, that this will increase a person's resting heart rate because they are no longer getting the signal to slow down the normal firing rate of the SA node. So both the sympathetic and parasympathetic divisions are controlled by cardiac centers in the medulla oblongata, which is part of the brain stem that we also covered in A&P 1. So in the medulla oblongata, there is a cardioacceleratory center, which is going to control the sympathetic nervous system and result in an increased heart rate. And there is also a cardioinhibitory center, which is going to control the parasympathetic nervous system and decrease the heart rate. Both of these centers in the medulla oblongata are responding to information they receive from baroreceptors, which are monitoring blood pressure, and also from chemoreceptors, which are monitoring the oxygen and carbon dioxide content of the blood. So the autonomic neurons are actually adjusting the heart rate by changing the membrane potentials in the pacemaker cells of the SA node. And your textbook goes into more detail that we aren't going to cover, but basically they're changing the permeability of the pacemaker cell membranes, which changes their potential, which either makes them depolarize more quickly, which would increase heart rate, or makes them depolarize more slowly, which would decrease the heart rate. Another factor affecting heart rate is the Bainbridge reflex, also called the atrial reflex. This is an automatic adjustment in heart rate based on venous return. And venous return is the amount of blood coming back into the atria from the major veins. So in the right atrium, blood is coming back through the superior and inferior vena cava. And in the left atrium, blood is coming back through the right and left pulmonary veins. So as the blood is coming back into the atria, it can stretch the walls of the atria, and that's what causes the Bainbridge reflex. So stretching of the atrial walls from the increase in venous return can activate baroreceptors in the atrial walls. This will cause an increase in depolarization of the pacemaker cells. It can also stimulate more sympathetic nervous system activity, which increases heart rate. So both of these things would then increase the heart rate. So basically, the more blood you have coming back, the heart's like, oh, we're getting a whole bunch of blood coming back. We better speed up our rate to get this blood pumped and moved on. Hormones can also affect heart rate. Epinephrine, norepinephrine, and the thyroid hormones T3 and T4 can all increase heart rate by acting directly on the SA node. So those were factors affecting heart rate. Now let's look at factors affecting the other part of the equation, which is stroke volume. So again, reminder that the stroke volume is calculated by taking the end diastolic volume and subtracting the end systolic volume. So anything that affects the EDV or the ESV is going to affect stroke volume. So we'll start by looking at things that affect the end diastolic volume. And again, this might help you if you think about this in terms of like how full a tube of toothpaste is before you squeeze it. And of course, in the case of the heart, we're talking about how full the ventricles get of blood after they're done relaxing. So the first thing would be the filling time. And the filling time is defined as the duration of the ventricular diastole. And this should make sense. If the ventricles are filling up while they are relaxed, then the more time they spend relaxing, the fuller they are going to get. So with a faster heart rate, you actually end up with a slower filling time, because as the heart rate increases, the time spent in diastole decreases faster than the time spent in systole. Another thing that affects EDV is preload, and preload is defined as the degree of stretching in the ventricular muscle cells during diastole when filling is taking place. So the blood is coming into the ventricles, and as the ventricles fill up, this is stretching the muscles in their walls. So the preload you can also think of as how full the ventricles are getting, so how much blood is going into the ventricles. So the preload would be directly proportional to the end diastolic volume. The more blood that gets in there during the filling time, then the higher volume you're going to have at the end of that relaxation period. So the more preload you have, the larger your end diastolic volume. And preload is affected by venous return, because if you remember, 70% of the ventricle's blood is actually coming in passively, and it's coming in straight from the veins, passing through the atria and into the ventricles before the atria ever contract. Go back and review the cardiac cycle if you forgot this. So preload is affected by venous return as well as how much time it has to fill, so the filling time. So basically the bottom line is the larger the EDV, the larger your stroke volume will be. And that should make sense. The more toothpaste you can get into the tube, then the more you can squeeze out when you finally squeeze. And this is also called the Frank-Starling principle. Basically, more in equals more out. The more blood I get in the ventricles, then the more I can squeeze out when I finally squeeze. All right, so now let's look at things that can affect the end systolic volume, or the ESV. And again, it might help if you think of this as what is left over after squeezing. So in our example of a toothpaste, this is the toothpaste that's left in the tube when you're done squeezing it. And in the example of the heart, it is what is left in the ventricle after it is done squeezing during a contraction. So preload can also affect the end systolic volume as well as the end diastolic volume. So the more stretched the muscles are in the ventricles up to a point, the more force they can contract with, and the more that they can contract, the more they're going to squeeze out, which will decrease what is left over, which is the ESV. Remember the length-tension relationship we talked about in Chapter 10 when we looked at sarcomeres in skeletal muscles? The same length-tension relationship also applies to cardiac muscle cells. And if you remember, an optimal sarcomere length will result in enough of a zone of overlap between the thick and thin filaments that the myosin heads are able to grab onto the actin subunits, but you still have enough room to shorten. So a graph that we looked at in Chapter 10 was the length-tension relationship graph, where we looked at if a sarcomere is too short, you don't get much tension. If a sarcomere is too stretched, you don't get much tension. You want this just right Goldilocks zone where you have just the right amount of overlap, but you also still have room to shorten, and then you can get the maximum amount of tension. Well, in cardiac cells, the pericardium, so remember we talked about the visceral and parietal pericardium, the pericardial membranes and the connective tissue prevent the cardiac muscle from overstretching, so we don't have to worry about it getting too stretched. And then preload is pushing it closer toward this optimal length. So preload is stretching those muscle cells in the ventricle walls just to the right point where when they do finally contract, you're going to be able to get maximum amount of tension, which means you're going to be able to squeeze harder. And the harder you squeeze, the less is going to be left over when you're done squeezing. So something else that affects end-systolic volume is contractility, which is the amount of force produced during a contraction. So again, this is actually a direct measure of how hard you squeeze. And again, the more you squeeze, the less that is going to be left behind when you're done squeezing. So the higher the contractility, the smaller the ESV. So contractility can be affected by the autonomic nervous system as well. So the sympathetic or fight or flight division increases contractility. The parasympathetic division, your rest and digest division, decreases contractility. Hormones and drugs can also have an effect on contractility. Epinephrine, norepinephrine, glucagon, and thyroid hormones all can increase contractility. Dopamine also increases contractility and is often given in emergency situations like in a patient in cardiac arrest. And you'll hear this on the medical shows as well. You'll hear them call for an injection of dopamine when a patient is having cardiac issues. Beta blockers are actually used to decrease contractility, and these are often given as prescription medications to treat high blood pressure. So by having the heart contract less forcefully, it reduces the pressure out in the blood vessels. And the last major thing that affects end-systolic volume is afterload. And afterload is defined as the amount of tension required to force open the semilunar valves to eject blood from the ventricles. So remember, the ventricles have to eject the blood through the semilunar valve out into one of the major arteries, either the aorta or the pulmonary trunk. And so the afterload is the force required to open that semilunar valve. So blood pressure in the arteries can have a big effect on this, as I'll show you in a moment. Also, damage to the semilunar valves can have an effect, like stenosis, like aortic stenosis, can make it harder to open the aortic semilunar valve. But let's look at blood pressure, because that's going to play a role in the average person's afterload. So imagine that you have a door and you've got two people pushing on both sides of the door. On one side, we have the ventricle trying to push open, and the door represents the semilunar valve. So the ventricle is trying to push open the semilunar valve so that it can get the blood out into the artery. But you've also got blood pressure in the arteries that are pushing back on the other side of the semilunar valve. So afterload is how much force the ventricle has to exert to overcome the pressure pushing back in order to open the semilunar valve and get the blood out into the arteries. An example is that vasoconstriction, when your blood vessels narrow, specifically the arteries, when the arteries narrow in their diameter, this increases blood pressure, and so that would increase afterload. It would put more pressure pushing back on the other side of the semilunar valve. And as afterload increases, ESV increases. So think about it. This means you have more left inside the ventricle when you're done squeezing. So basically, you are spending so much time trying to force the door open that once you get it open, you've got actually less time to pump the blood out. So more blood gets left behind in the ventricle, which is going to increase the end systolic volume, which is what is left behind after squeezing. So here's a summary from your textbook that summarizes all of the factors affecting the stroke volume. So we've got factors affecting the end diastolic volume over here on the left. So you've got preload, which is affected by venous return and filling time. And then we've got factors that affect the end systolic volume on the right. And again, we have preload, we also have contractility, and then we also have afterload. So our last section is a summary about the cardiac output, and then we'll make one final mention of a new term called cardiac reserve. So here is the picture from your textbook. And again, cardiac output is defined as the heart rate times the stroke volume, so the amount of blood pumped out of a ventricle in 1 minute. Heart rate is affected by things like the atrial or Bainbridge reflex, innervation by the autonomic nervous systems, and hormones. Stroke volume is described as the end diastolic volume minus the end systolic volume. Things that affect the end diastolic volume include preload. Preload is affected by venous return and filling time. End systolic volume is also affected by preload. They forgot an arrow. It's also affected by contractility and afterload, and afterload can be affected by vasodilation or vasoconstriction. All right, so all of these things can overall have an effect on your cardiac output. So one last definition for you in this chapter. A cardiac reserve is the difference between your resting cardiac output and your maximal cardiac output. So like your resting cardiac output is your cardiac output when you're sitting on your couch watching TV. Your maximal cardiac output is how much your heart can pump out in 1 minute, like when you're doing heavy exercise. In a normal person that's, not very athletic, there is a 300 to 500% difference, which means your cardiac output can be up to 500 times greater when you're working out or doing heavy exercise versus when you're just laying around. Athletes who have a very well-conditioned heart can actually have a cardiac reserve of up to 700% difference, meaning that at their peak exercise levels, their heart can actually pump out 700% more than it does when they are at rest. So training and doing cardiac exercises to improve your cardiac endurance can really help to increase your cardiac reserve. Note that normally as your heart rate goes up, cardiac output will go up, but once heart rate goes over 180 beats per minute, cardiac output will actually decline because the stroke volume starts to get very small. So when the heart rate gets too fast, there's not enough filling times for the ventricles to fill up with blood, so the stroke volume becomes very small, and overall that's going to cause a decreased cardiac output. And then that is the end of chapter 20. We'll finish up the cardiovascular system in chapter 21.